This is Dr. L, and we're going to talk about some of the basics of cell walls. Let's think about what cellular organisms have cell walls. So you should be familiar by now with the three-domain tree of life, a way of mapping all living things past and present. There are three domains: the bacteria, the archaea, and the eukarya or eukaryota, as some describe this. At the very base of the term, you'll see the term LUCA, which stands for Last Universal Common Ancestor. That's the point at which we see the trees starting to branch into these three groups. The bacteria and the archaea contain all of the prokaryotic cells, those cells that lack a nucleus, and the eukaryotic domain contains all of the eukaryotes. Humans fit into the animals, along with like your dog, etc., etc. When we look at the three-domain tree of life and we think about whether cell walls exist in the cells of these organisms or not, what we know is this: the vast majority of all bacteria have cell walls. I think all the archaea that we know about so far have cell walls. And when we look at the domain eukarya, the vast majority of organisms in that domain also have cell walls. So while humans lack cell walls, and so do other animals, all of the other organisms within the domain eukarya do have cell walls. So really the norm is to have cell walls. Those cell walls can differ in their chemistry, but there are also a lot of similarities in the chemistry and in the function of cell walls, and that's what we're going to talk about next. In any cell, the location of the cell wall is outside of the cell membrane. Okay, so the cell membrane lays on the inside, bounding the cytoplasm in the cell, and then outside of that structure in green here is the cell wall. So the cell wall will always be found on the outside of the cell membrane. The major function of cell walls is to provide structural support for cells, mainly to protect them from something called osmotic pressure or osmotic stress. This is the pressure created by water moving across a membrane due to a process called osmosis, which will be addressed in other locations, or you can look up. Okay, so it's protection from water pressure, too much water or too little water. That's why we need cell walls. They provide structural support and they protect us from osmotic stress, and they are largely made out of structural carbohydrates. Okay, so carbohydrates are one of our four categories of macromolecules, the others being nucleic acids, lipids, proteins. And carbohydrates can be either structural or they can be for energy storage or energy provision in a cell. And the carbohydrates that make up cell walls are for structure. Carbohydrates, like all of the macromolecules, have building blocks. They are called monosaccharides. Two examples of monosaccharides are shown here. Glucose, D-glucose being one of the forms of glucose, and ribose, D-ribose being one of the forms of ribose. The arrow here shows how a glucose can exist in a linear conformation or in this ring-like alpha-D-glucose structure shown on the right. And what will happen is there'll be some toggling back and forth between those forms. The same will happen for any of the monosaccharides. So they exist in their basic chemistry, and then they can assume different three-dimensional spaces, as shown here. Carbohydrates, the monosaccharides that make up carbohydrates, they can vary in the number of carbons. And so glucose contains 6 carbons. Ribose contains 5 carbons, which is to be a pentose sugar. You'll see ribose in some of the nucleic acid building blocks, so we'll see that again in the future, and we'll mostly look at it drawn in its like ring-like conformation shown here. The light green numbers indicate the number of the carbons. So the 1 is carbon number 1, the 2 is carbon number 2, the 3 is carbon number 3, the 4 is carbon number 4, and then the 5 is carbon number 5, and there's also a C drawn here, whereas elsewhere in the molecule, we're just inferring that that's a carbon because covalent bond lines are connecting together, which always means there's a carbon present. Okay, carbohydrates and the monosaccharides that they're made out of tend to be very polar because they tend to have a lot of oxygen bonded to carbon, and a carbon-oxygen bond is a polar covalent bond. The electronegative oxygen will pull those shared electrons toward it, creating areas of partial charge. This means carbohydrates, in general, can form hydrogen bonds with water. They're very hydrophilic. The chemical bonds that hold monosaccharides together into carbohydrates are covalent bonds that have a special name. They're called glycosidic linkages. Here we see the monosaccharide glucose and fructose, and an arrow showing the chemical reaction where we're forming a glycosidic bond, which is shown there at the bottom, holding these two monosaccharides together. You can have alpha glycosidic bonds and you can have beta glycosidic bonds, and we won't get into that here really, but the alphas tend to be relatively easy to break down. The betas a little bit less so. Structural carbohydrates will tend to have more beta glycosidic linkages or bonds, whereas energy-based carbohydrates tend to have more alpha. Carbohydrates serve a variety of functions in the cell. They're there for energy, so glucose, for example, can be broken down directly to make ATP. They're also present sometimes for energy storage, so think starch in a potato. It's a storage molecule. They're there for structure, and that's their role in a cell wall, so think cellulose or peptidoglycan, also a structural carbohydrate. And carbohydrates also play an important role in something called cell-cell signaling, like identifying what a cell is so that other cells know. And they're usually found as fusion molecules attached to a protein or something else. And they play other roles too, but these are the major ones. So in plant cell walls, one of the major structural carbohydrates is the chemical known as cellulose, shown at the bottom. And cellulose is a polymer of glucose, formed using beta-glycosidic linkages. So each of these little ring-like structures was a monosaccharide, and they're connected together by multiple glycosidic linkages. These are very hard to break down. Only a few different types of microbes have the ability to do this, so we can't break cellulose down, which makes it a great structural carbohydrate. Like we as humans lack that ability. That cellulose will organize into structures called microfibrils, which organize into larger fibrils, which extensively cross-link with other molecules like peptin to form this complex rigid structure that makes up the cell wall in many different plants on the planet. Bacteria, in the domain bacteria, we see a lot of cells, they have the ability to make a structural carbohydrate called peptidoglycan. Only the bacteria can make peptidoglycan. And this is a drawing, one of the ways to draw peptidoglycan. Each of the little like 6 hexose-shaped structures is a carbohydrate. There are different ones there, and I won't get into the chemistry of those, but they're very similar to glucose, okay? And then each of the little balls are amino acids, different types of amino acids. So you might be thinking, wait a minute, amino acids are in proteins. Well, amino acids are also in peptidoglycan. So this is an example of really a fusion molecule. It's not strictly made out of carbohydrate. It's carbohydrate, which has monosaccharides and glycosidic linkages, yes, but there are strands of that are cross-linked together with chains of amino acids, which are called peptides. Hence, peptido and then glycan. Peptido for the peptide portions and glycan for the sugar or carbohydrate portions. In bacteria, there are different ways of organizing the peptidoglycan. So this drawing here on the left is showing 2 types of bacterial cell walls, gram-positive and gram-negative. And this relates to a lab that we did in class, or that if you're not in my class and you're watching this video, labs that happen in a lot of introductory biology and microbiology classes. Gram-positive bacteria have a cell wall that's made of a big, thick layer of peptidoglycan, and gram-negative bacteria have a thin layer of peptidoglycan, and then outside of that, a space called the periplasm, also inside of that, a space called the periplasm, and outside of that, something called the LPS, or the endotoxin, or more commonly the outer membrane. LPS stands for lipopolysaccharide. I will tend to use the terms outer membrane or endotoxin. Endotoxin because often this outer layer can be toxic to humans when found in a gram-negative pathogen. A lot of the molecules that exist in all cells are these fusion molecules, by the way. And so while we're talking about peptidoglycan being a fusion molecule, let me point out, taking you back to cell membranes, that many of the molecules in cell membranes are also fusion molecules. So here we see a carbohydrate tagged onto a protein. We call that a glycoprotein, glyco for sugar, protein for protein. And here we see a carbohydrate tagged onto the top of a lipid. I don't know if it's a phospholipid or more likely it's some kind of a steroid lipid. When we see carbohydrates tagged onto lipids, we call them glycolipids. So those are two examples or two generic names for fusion molecules. When we see these in cytoplasmic membranes, they're generally playing a role in cell-cell signaling. Back to that gram-positive and gram-negative piece, in a gram staining lab, you can take different kinds of bacteria and you can do a chemical staining process called gram staining, developed by Christian Gram back in the mid-1800s. And gram-positive bacteria will show up as purple or dark, and gram-negative bacteria will show up as pinkish. Right? And a cross-section of those gram-positive and gram-negative cell walls, another cross-section, here's the gram-positive, thick layer of peptidoglycan. Here's the gram-negative, thin layer of peptidoglycan, and something called an outer membrane, whose chemistry I'm really not going to get into, but it's a little bit similar to the cytoplasmic membrane. These will stain pink in the gram stain, and these will stain like purple or dark blue, sometimes almost black looking through the membrane. And that concludes my discussion about the cell walls for this video.